I have been practicing what I teach. We just talked about viral evolution, exchanging viruses. So I exchanged viruses with someone. So I've got reassortment going on within my own body at the moment. So I'll use that as an excuse for, yet again, forgetting to activate the microphone. Okay. So our main focus today will actually be on topological landscapes for visualizing scalar valued functions. Basically, we will be taking a look at RNA folding, but one can also take a look at energy folding, protein folding. And one frequently wants to visualize the energy landscape. And so that's what we'll be focusing on today, is visualizing that energy landscape. On Friday, we will review the MAPR software applied to RNA folding. So today we will start off with a very elementary introduction to RNA. So RNA is very similar to DNA. So on the left side, we see basically pictures of RNA, and on the right side, we have DNA. They both have a sugar phosphate backbone, so we've got a sugar phosphate backbone. They both have bases, but with DNA, you've got base pairs. You've got a nice double helix, versus RNA is sometimes single-stranded, sometimes double-stranded, and we'll see some examples of that. One of the main differences between the two is DNA is deoxyribose, so there's just a hydrogen here within its sugar atom in the sugar phosphate backbone, versus here it's an OH group instead, and that makes it more reactive. And the other difference is they both have cytosine, they both have guanine, they both have adenine for base pairs, but DNA has thymine versus RNA has uracil instead. So we will see when we do have base pairing, G will usually pair with C, and A will usually pair with U in terms of the RNA base pairing. When we think about, I moved a slide, let me see. Okay, sometimes I accidentally move slides. When one thinks about RNA, one frequently thinks about messenger RNA. That's the central dogma of life. We have our DNA. Our DNA gets pulled apart, so we've got the DNA here. That allows for an RNA copy to be made. And so then we've got our RNA copy. If we're a eukaryotic, it then goes outside of the nucleus to then get translated into protein. So we've got our RNA that then will start getting transcribed into protein here. So we've got our DNA to our messenger RNA into our protein. So when people think about RNA, they often think about the single-stranded messenger RNA version. They assume it's single-stranded. It's not always single-stranded in regions, but in order to get transcribed, this part here has to be single-stranded, just like when you transcribe your DNA, your DNA has to be made single-stranded to work with that, and I pressed the wrong button. So that's often what we think about, and so that means that one of the functions for RNA is to store genetic information. So it does store genetic information just like DNA does, but being, this structure with that OH group, it's far more reactive, so it's not as stable. So it is an unstable method for storing genetic information. Your RNA will get degraded, so there will be proteins that will eat it from the two ends, versus your DNA is more stable and doesn't usually get eaten, but there are times when it gets eaten as well. Any questions about that? But you'll notice that there's this thing called tRNA. tRNA also has functions just like protein. So this is your transfer RNA, and you can see that we do have a number of base pairs happening right here. G pairing with C, A pairing with U. There are some single-stranded regions as well, but this is the basic structure for your transfer RNA. When we talk about structure, there's actually 4 different things that we can mean. We can first off talk about the primary structure. The primary structure would just be the sequence. So if our RNA sequence is A, U, G, C, C, that would be your primary structure, is just the sequence. We then have secondary structure, and that's basically telling us this base pairs with this base pairs with this. Now, while I'm using the example of RNA, we have exactly the same thing happening with protein as well. Acids contacting each other, possibly through hydrogen bonds, possibly through covalent bonds, normally hydrogen bonds, but you do have some connections. So we've got hydrogen bonding between the base pairs in RNA, and hydrogen bonding, and on rare occasions, covalent bonding between the amino acids for this or protein. So our secondary structure just tells us what are the things that are connected. And so sometimes if you do your sequence, you'll just say, oh, this connects with this connects with that, and that would be saying that we're bringing these base pairs together. We then have the tertiary structure, so the tertiary structure, and that's the actual 3-dimensional picture. So now we've got the 3-dimensional picture as to how the RNA folds. And then after that, there's the quaternary structure, and that's when things interact. So we might have RNA here, we might have a second RNA interacting with the first one, we might have some protein interacting here as well. So the quaternary structure is when you have more than one complex so more than one RNA or more than one protein together, forming a 3-dimensional complex. Any questions about that? So we have our transfer RNA, and that basically just brings an amino acid. So we've got our amino acid here, that it then brings it, and then it gets hooked on, and so you've got your sequence being created of amino acids forming your protein. But RNA has even more functions. Right here, where this thing happens, where the messenger RNA gets transcribed into protein, that happens in the ribosome. The ribosome is about 60% RNA and about 40% protein. And so if we just take a look at the RNA part, here we see on the top part, that's our secondary structure, showing how part of the ribosomal RNA, this is actually the small, there's a small and a larger one, and so I think it's this blue part here where you see the secondary structure, and then this is a different picture of the tertiary structure where we now have sort of fill out the atoms and they take a bit more space, and so this is the RNA part of the ribosome. So RNA has many, many different functions. But before we talk about the many different functions, we can also sometimes you'll see these circular plots. So you'll see a circular plot, and all that means is that if I see where the 0 is, so the 0 is someplace around here, what we're doing is we're drawing the RNA sequence. So this is just your RNA sequence just being drawn as a circle. And then we have the circular plot saying that this base pairs with this base pairs with that, and that's where we get all these arcs from it. So if we take a look at this ribosomal RNA, I believe the starting point is right here. Starting or ending, I don't remember which, but we'll just say it's A, U, lots and lots of other things, and then it ends with a U. And so if I were to notice that this base pair is over here, so we just draw an arc to represent this base pair, but I have this big long sequence in between. It might be easier to draw this on a circle. So we've got our A, U, our U. Often there is binding for the next things in between, and so we'd have lots of things representing this binding right here. And then as we go along in the sequence, these parts are not base paired, but these ones here, they are base paired over, sorry, these are base paired. But there is a gap in the middle, and so you'd have in sort of, so you'd have for this region here, you'd again have some base pairing. That's not very good. Let me redraw. We'd have some base pairing for those 5 things here, so we can draw the 5 circles to denote this base pairing. Then there is this gap where on either side nothing is paired. And then we have some more base pairing, so some more base pairing, and then a gap where nothing is paired again. And so that is this circular art plots that you might see that will illustrate the secondary structure of RNA. Any questions about that? We can also take a look at a pseudoknot. So in this pseudoknot, if I were to draw this as a, let's do this as just this one instead of the circular one, either way works, but we can see that the U is not paired, but then the C does get paired with some things over here. So we've got a whole bunch of base pairing. Eventually there's this gap here, but the main thing is if I go along here, I've got all of these, so we encounter this first. Then we've got a big region that's not base paired. As I go along here, I now get this next, so region 2 would be next in my sequence, and so that also gets base paired with a number of things. And then as I travel along, I get to region 3, so that's region 3 over here. And as I continue going on, I then get to this region 4 right here. And so we can see in terms of our sequence, region 1 connects base pairs with region 3. Region 2 connects with region 4. And so we've got this sort of pseudoknot. They call it a pseudoknot when these things cross. So sometimes in your circle, we had circles where they didn't cross, other times where they do. And so here there's no pseudoknots in the middle column or the right column, but you can see that there is some crossing here. And so we do have some pseudoknots in these types of situations. Any questions about that? So RNA can form very interesting structures, and it can do things. So RNA, there isn't just the messenger RNA. Yeah, there's messenger RNAs, but there's transfer RNAs, there's tRNAs, there's all sorts of different RNAs. So here's a nice little list. Yeah, messenger RNAs is one thing. So they do encode for proteins. They are temporary storage of genetic information. But in many other cases, they're very active. So remember when we talked about the viruses? Those viruses actually had proteins within the RNA sequence. So they coded for proteins within the virus RNA sequence so that they could then make more copies of itself without going through DNA at all. So RNA has a thing that acts, it's active in many ways similar to proteins. They have three-dimensional structures similar to proteins. It's base pairing instead of, amino acid contacts, but they do have a lot of similarities to protein. They do have actual structure, and that structure is related to their function. So just like for proteins, you've got a structure that implies a function, and they're very active in doing things. And there are all sorts of different types of RNAs. So one thing that you might hear sometime is about the RNA world. And in the RNA world, it's basically thought that one way that life evolved is that RNA came first. RNA stored information, but it wasn't very good to store. It would degrade, and so therefore DNA came about, but we still do have RNA viruses. If we start off with RNA, well, the RNA also had functions, so it could act like proteins, but then eventually proteins could do more things, had more flexibility, and so eventually they evolved. And so if you think about these viruses that have, some of which have RNA genomes, and the ribosome and a lot of other, you've got a whole bunch of things that actually perform functions very similar to how proteins act. So that's one hypothesis, is the RNA world. But the main thing to keep in mind is that RNA has both this temporary storage thing, similar to storing genetic information like DNA, but it also has functions similar to protein. Any questions about that? So now let's talk about the Harvey and Wong paper in preparation for Yusu Wang's talk on Monday, December 9th at the IMA. One thing I want to point out is that I'm talking about a paper that was published in 2010. She will give the update 3 years later. And so when I say that if I mention any limited functionality, well, maybe that limited functionality no longer is limited, so there might be a lot more. Well, we'll have to be in suspense until Monday, December 9th to find out what the additional functionality is. But let's start off with the motivation. The motivation is basically we'd like to understand protein or RNA folding, and one frequently takes a look at an energy landscape. An energy landscape is just a function that goes from your set of conformations. So here is an example of a conformation. And then one can calculate the energy of this conformation. And so we get out some real number that states what its energy is. If things are high energy, then it's less stable. If it's low energy, usually proteins try to fold into their low energy state. The typical way, so this is a very typical picture. You'll see similar pictures in lots of biology textbooks, is you've got your conformation space. So maybe this is, one, particularly folded protein. Maybe this is another partially folded protein. Down here would be your your lower energy state, where it would be you expect your protein to not be quite as folded up here, more folded as it comes down here, and eventually it folds into its fairly stable state into the low energy. Now, one thing to keep in mind, things are not static. I forgot who gave this analogy, but someone told me once that a crystal structure of a protein, so the static image, which is what we see, the picture on the preceding page, could have come from, actually that was coming from a simulation, a computer simulation of folding, but it could also have come from a crystal structure where you take a snapshot. But at any rate, whenever you have a snapshot, if you want to understand a protein via a snapshot, it's very much like taking a picture of a horse in a barn. If I want to bet on a horse to see which one's going to win the race, and am I going to be basing my bet, how good of a runner the horse is, by a snapshot of the horse in the barn? That's not exactly, it doesn't really tell you how the horse runs. It doesn't tell you how the horse functions. Yes, it's a pretty picture. You can have pretty pictures of a horse. Yeah, my pretty picture of a horse. just like we can have pretty pictures of, these protein structures. But it is a very static snapshot, and it really doesn't tell you much about how it functions. Now, there are some structural proteins that may not move that much, but many proteins, they are not as static as we often visualize them. But that being said, they still like to fold into a fairly stable, there's still some movement in there, but it still likes to fold into a fairly stable, low energy conformation. Any questions so far? So back to our picture again, we basically have this represents a conformation, this will represent a conformation, and we can map this over via our energy function to the real line. So anything on this contour right here will map over to this, fairly high energy. Anything in this region will be mapping over to some lower things. And then over here, we get mapped to even lower energies. So we've got this energy function that maps from our sort of energy landscape that we'd like to visualize. So we'd like to sort of visualize the graph of here we've got our conformations, and here we've got our energies. And so if I take this conformation right here, say it maps to this energy, and so we can plot that. And that would help us visualize how our protein is folding. Any questions there? Okay. So, however, notice that all my conformations, the energy is the third coordinate. My conformations here, I'm just using two coordinates. Once I specify my x and my y, I can map it up to whatever its energy is and do my, coordinate 1, coordinate 2, energy. And so I'm just using two coordinates to identify my conformation. In other words, this is not a real energy landscape because there is no way one can represent a real protein conformation using only two coordinates. You need way more coordinates. And so in reality, we have something where our function, our energy, is going into some higher dimensional space, and then we output its energy. And if we want to take a look at the graph, then the domain would be RD, the codomain is R, and so we'd be trying to visualize R to the D plus 1. And if D is greater than 2, that's hard to visualize. And generally speaking, it's greater than 2. So in the Harvey-Wong paper, they basically analyzed data from Park and Lee. They created 20,000 conformations via molecular dynamics simulation, and they focused on a backbone of 46 alpha carbons. So the data that Harvey and Wong were simulating was actually pairwise distances between these 46 atoms. So in other words, there's a matrix where you've got these 46 atoms here, the 46 atoms here, and then this element is the distance between atom I and atom J. And we only need to know the upper distances because it will be symmetric. The distance from atom I to atom J will be the same thing as from atom J to atom I. They'll be exactly the same number. And so we get 46 choose 2 values. And so the original distance, the original conformation space that they were given that describes the backbone was this pairwise distance. And they had 1,035 numbers to represent this pairwise distance. And so they had a representation of the backbone in R to the 1035, and then you can map that into R. I don't know about you, but I have difficulty visualizing graphs in R to the 1036. So one can, of course, do much better than that. Instead of taking a look at the pairwise distances, one can instead take a look at the 46 alpha carbons, and each of them come with an XYZ coordinate. And so we've got 46 times 3, so that is 138 possibly. And so we could be looking at the set of conformations, so maybe S would be a subset of R to the 138, where the first triple would correspond to the first alpha carbon, the second triple would correspond to the 3D location of the second alpha carbon, etc. One can then, of course, mod out by translations and rotations any rigid movement of this. If I rotate it or I translate it, I will get exactly the same protein. And more than that, you don't need all 100. It's not. It might live originally in 138-dimensional space, but a circle. If I draw a circle lives in 2-dimensional space, but I really only need 1 dimension. The angle to describe the circle. So I can describe a circle using only 1 dimension. So a circle is 1-dimensional, even if we have to draw it in at least a 2-dimensional space. This particular protein, we're looking at this survivin protein. It's been estimated that it's actually about a 20-dimensional space. So you've got a 20-dimensional manifold living into some higher dimensional space, whether you want to take that higher dimension to be 1035, 138, or something smaller after you mod out by translations and rotations. In either case, if we've got something that's around 20-dimensional, so we've got a 20-dimensional space, I still can't visualize 20-dimensional spaces very well, and so we'd like to project down to smaller dimensions. Any questions about that? So this is one method. Basically, we project onto 2 dimensions. So we have here, so we're now going to project our conformations, our set of conformations, to lie into some 2-dimensional space by taking the 2 first principal components. So principal component projection, projecting onto 2-dimensional spaces. And then to get the energy, we can color code. So this here is a subset of R2. So I've got a point in R2, but we color code the energy. Blue is low energy, and then red is high energy. So that's one way of trying to visualize the energy landscape. But out of my expected 20 dimensions, I'm only seeing 2 of them, and it looks very rough. And you get all sorts of, it looks more rough than it really is. You can imagine if you want to understand a 3-dimensional object, it's nice to see it in 3 dimensions as opposed to projecting it into 1 dimensions or 2 dimensions. But we can't see things in, higher dimensions, and so we do have to do some projecting. Is there a better way of doing this? Here, by the way, is the rotation, and so this is the same picture as on the preceding page, but now our energy is now the z-axis, and our conformation space is here in R2. And so you can see the low energy and the high energy for different conformations. But again, projected our conformation space into 2 dimensions. So another way to visualize these energy conformations is via contour trees. Contour trees have been around for a number of years, from at least the 1940s. It's similar to the Reeb graphs that we discussed previously, where we basically just take the pre-image. So if I take a point here, I take its pre-image, and then I collapse the connected components. I collapse the connected components. I've got two connected components right here. Collapsing those. We could get our contour tree or Reeb graph if our function is nice. And so this we can think about, what persists as well. Here we've got our component containing B, our component containing A, B and A, and eventually they merge into one component, and so eventually they merge into one component. So very similar to what we've done before with respect to persistent homology. So that's an old method, and it actually works fairly well. Even if this were in, R to the 20 or so, you can still do this sort of thing where we still take the pre-image of a point. We can still take the pre-image of a point and take a look at all the connected components and do this sort of merge process. So that works quite well, but it'd be nice if we could encode additional information. So one thing to just note is what I will be talking about today, we will only look at trees. One can also get in this Reeb graph here, here we've got one component, now we've got two components, and then we go back to one component. So we had one component, it separated, it split into two components, and then it merges into one. And so we can get these Reeb graphs that are more general, that are not simply connected, that have these non-trivial H1, so H1 is not 0. We are not going to look at that example. I don't know if Yusu Wong will look at that in December or not, but today we are going to focus specifically on trees. So our output will be an actual tree, and so we won't have any interesting topology here, so no holes in our shape over there. Any questions so far? Okay. So we can now, so we've got our level sets. If I take, a point here, I take its pre-image, and that gives me my level set of F inverse of R. A contour is if I take one of these, it will project to, say, a point Q, and so then C of Q is the connected component. So if I have a P over here, this would be my C of P. And then what we want to do is take a look at the topocomp of an edge is just the union of all these contours within that edge. So if I take a look, for example, at this edge right here, I would have contours for this, contours for that, and so my topocomp of this edge, so if I call this edge E1, the topocomp of E1 is the union of all these contours, and so we would get this entire area right here. So we'd be getting all of this area right there. One way to get additional information is the area of this. So we can say that the area of this thing here will imply the length of the edge. So we can get some information at least about what our conformation space looks like. It's important to note that, going from here to here, there's a bigger well than if I'm going from C to A. Here there's a smaller well right here. So this blue edge gets mapped back to the topocomp. So if this is E2, this would be topocomp of E2. And it has a smaller area, so we have a shorter one. The brown edge, if that's E3, then that goes back to the topocomp of this E3. And you'll note that in some cases, the topocomp is this nice disk, we basically just took a disk and then punched it, or in this case, we have something that is, has this hole and this hole here, and so it might look like that. But in either case, whatever area we have here, we'll then decide that that length is going to be the same. Now, I mentioned area, but I don't really mean area. For this particular example, this is mapping back, this topocomp maps back to a surface, so it maps back to this brown surface right here. So it maps back to that surface, so I have area. But in reality, remember, my energy function is going from some d-dimensional manifold into R. So when I map back to the connected components, I'm getting d-dimensional connected components, and so I'm actually going to be getting volume. So what we want, basically, is given this map from this d-dimensional manifold where our surfaces are okay. So I think now you can hear and see things, maybe. So you can hear okay. Okay, good. So sorry, I checked on Wednesday for an Ethernet cord, and I forgot all about plugging it in and didn't notice that it was missing until I needed it. But hopefully this will work now. So you can imagine creating this. So the idea, again, is we have a function. I need to select the pen. We have a function from some, well, d-dimensional manifold into R. We'd like to instead create a function that keeps a lot of the information from R2 into R, because we can visualize these graphs a lot better. But we'd like them to have exactly the same contour tree, but it's nice to have something more than just the contour tree. We can use the lengths of the edges to tell us to correspond to the areas of, so if I map this back, this maps back to this area right here. We could use the length of those edges to map of it. But an easier way, if I want to, represent this, why not just represent it by an actual area? So we can plot our points in fairly arbitrary spots, so just arbitrary plot E, A, and B. And then this edge has a particular area, so we just choose some conformation of the same area as this. Then we can do the same thing for B, the same area. We just need to make sure that they join at C, because they do have a point C in common. And so we can map, choose each of these contours. And so we're still choosing a map into R, where the point B gets mapped to whatever energy it was before. The point A gets mapped to the energy that this point was. Anything here gets mapped to the energy over here. Anything over on here, anything here will get mapped to the energy over there. And so we can come up with creating a new contour map that lives in R2. So choosing these contours to go to the same energies as wherever these contours come, but creating something with exactly the same surface area. Similarly, we can do the same thing for E. We just choose a bunch of contours around here so that by the time I get to D, I have the same. So even before, if I take this point and I map it to this contour right here, then I want this area to be the same thing as this area right here. And continuing on, we can then say that we want to map this thing here to a bunch of contours. So we can map it again to a bunch of contours, just making sure that the area it encloses is the same. until we get to this thing right here, and then we can continue going up. For here, this contour should enclose a certain area, and so this contour would be mapped to, whatever distance that this point here was mapped to. So that is a way of coming from your function, your scalar valued function from some d-dimensional manifold into R2, where instead of this being area, this might actually be volume. So in this case, we went from d equals to 2, so we looked at an example for d equals to 2, and note that my example of getting this R2 space right here, if I were to draw the graph, if I were now to draw the graph with my R2 and my f of R2, I would actually be getting this function back again. I'd be getting exactly the same function back. And so you can see that if we do the case d equals to R2, we get basically the same thing. There actually could have been some distortions because we don't really know where E, A, and B is, but it would be something that would look very similar to this type of thing. Yeah, it wouldn't necessarily be identical. Maybe we accidentally did B and A instead, but it would be an energy landscape that would be similar. Versus if we now take a look at, D being much greater than 2, then instead of having the areas match up, we now would be having volume. And so this point here would be getting mapped to, its contour would be some type of volume here, and we would have the volume match up with the area. And so that is one way of going from, instead of trying to visualize a graph in RD cross R, we can instead look at a graph in R2 cross R, where there's some similarities between the two graphs. The area for this one will match the volume of the pre-image for each of the contours. Any questions about that? So we just need to have a way of coming up with that, and the way to come up with that she actually uses squares. There's other ways of doing it as well, but if we know that we have, we'll draw the arrow. So the arrow means that the component of CV will be contained in the component of CU if I've got an arrow that points from U to V. So I've got an arrow that points from U to V. And in terms of the energy landscape, we just need to say maybe this is a leaf. So we might have this leaf right here. Maybe it's a local maximum, or maybe it's a local minimum. And so maybe when we're doing the graph, we would actually be having for the local maximum, it'd actually be doing some sort of, thing that I can't really graph this sort of thing, but it might be where if I were to draw the contours, we'd be having different contours, and this would be stacking up, or it might be stacking below. So if it's a local max or local minimum, we can still choose these squares instead where they max up with the volume. But we have gone over time, so we will have to finish this up next time. Any questions about anything today? So see you Friday.